तू पाणी ठेवू शकतो तिथे. सॉरी, पाणी तुझ्यासाठी. तुमच्यासाठी एक आहे, मी घेतलं. पाण्याची गरज आहे.
Hello, hello.
असं काय?
Sorry, excuse me, everyone. Could I please request that you put your phones on silent for the graduation ceremony, please? Thank you.
अब नहीं आएगा, अब मेरे को ही दिख रहा है. थैंक यू. ये थे.
Ik open de zitting van de commissie ter beoordeling van het proefschrift en de stellingen van Sumit Moreshwar Meshram. De verdediging zal in het Engels worden gevoerd. A warm welcome for all of you at the public defense by the candidate of the thesis entitled Design of Biodegradable Soil Moisture Sensor Powered by Bacteria Charged Paper Batteries. The Board for Doctorates of the TU Delft has installed a committee to evaluate the thesis and propositions. The committee will have a debate with the candidate in public. It is an honor to present the members for this committee, to whom I extend a hearty welcome. First of all, on the opposite table, closest to the audience, we have Professor Arrevind from the University of Groningen in the Netherlands. Professor Arrevind, thank you so much for coming to Delft. We very much appreciate it when professors from other universities travel to Delft for a thesis defense. Next, we have Professor Strik from Wageningen University and Research in the Netherlands. Professor Strik, also thank you so much for coming to Delft. It's very much appreciated. Third, next to Professor Strik, we have Professor Van Lier from the Faculty of Civil Engineering and Geoscience of Delft University of Technology. And finally, on the opposite side of the table, furthest away from the audience, we have Professor Uilenhoed from the same faculty of Delft University of Technology. On this side of the table, furthest away from the audience, we have the co-promoter, Professor Pande from the Faculty of Civil Engineering and Geosciences of the TU Delft. On my left, we have Professor Jourdain from the Faculty of Applied Science, who is also a co-promoter. And on my right, we have Professor van der Giese from the Faculty of Civil Engineering and Geosciences of the TU Delft, and Professor Giese is the promoter today. Thank you all for taking part in this academic ceremony. My name is Vlucht from the Faculty of Mechanical Engineering, and it is my pleasure to share this session on behalf of the Rector Magnificus. To start the defense, I kindly request the candidate to go to the lectern together with the paranibs. Waarde promovendus, I kindly ask you to give a short presentation on the content of your thesis, understandable for non-experts. You should highlight the results obtained, their relevance, and possible applications of your findings. You have exactly 10 minutes for this.
Good afternoon, Chairperson, external committee members, my promoters, colleagues, and my dear friends. Today, I'm going to talk about my PhD dissertation topic, design of biodegradable soil moisture sensor powered by bacteria charged paper batteries. So why there is a need for designing this kind of a batteries? So in the rural areas of the developing countries, the farms are small and they are located in the remote locations, which do not have the electricity grid. Because of the lack of the electricity grids, the farmers from these regions, they cannot use the available irrigation practices to irrigate their farms. So they are dependent, so they use traditional unscientific irrigation methods to irrigate their farms. Because of that, this unscientific method, there are water loss from these fields. On the other side, because of the climate change, some regions in the developing countries, they are getting floods and some regions are getting droughts. If these farms are located in the drought-prone regions, there is a need to use the available water effectively. So there is a need to use this water in an effective manner. So there is a need to there is a need of such kind of a device which can help us to manage this water in an effective manner. So using soil moisture sensor, we can reduce this water loss and we can use the water. We can do the water management effectively. But the farmers or the smallholder farmers cannot use this kind of soil moisture sensor because, as I told you, there is a lack of grid. And these available soil moisture sensors are expensive to the smallholder farmers. But there are some farmers who are rich, they can use these soil moisture sensors which are powered by solar energy and by the batteries or other renewable energy sources. But these solar energy and the batteries, they are toxic to the environment. They are non-degradable and they produce the e-waste. So in order to solve this kind of a problem of off-grid nature, cost effectiveness, and the environmental unsustainability, there is a need of designing a low-cost off-grid biodegradable soil moisture sensor. So how this soil moisture sensor will work? So first we put this whole system into the soil. As we put the system into the soil, the bacteria that are present in the soil, they will come and attach onto the outer surface of the battery. When we put more water, more bacteria will come and it will emit electrons and will produce electricity. And there is a tag embedded in this system, so which will emit the signals, and that can be captured by the mobile phones. And through mobile phones, we can sense how much soil moisture is present in the soil. So what are the important concepts that I use in these things? So first concept is about the conventional battery. So conventional battery is a device in which the chemical energy is converted into the electrical energy using the redox reactions and using the redox reaction. Next is the soil microbial fuel cell concept. So in this, this is a technique in which the bacteria that are present in the soil, they emit the electrons, and these by oxidizing the organic matter present in the soil, and then after emitting the electron, they produce the electricity. So the third concept is about the paper battery. So the paper battery is a thin, flexible device which uses paper as a substrate. And by using papertronics and the origami techniques, we designed this paper battery to power the small electronic devices. So this is the whole system by using, which we have designed using the concept from paper battery, soil microbial fuel cell, and conventional batteries. So what are the important parts that we have to take, we have to design while designing this whole system? So you can see this is a 3D model of my system. So the first important part is the anode. So anode is the which releases the electrons. So the black color, the color is the anode. Second is the cathode, which gains the electron that are produced onto the anode. So the orange color is the cathode in my system. Third is the membrane. So membrane is a part which separates the anode and the cathode. So the part between the anode and the cathode is the membrane. Then fourth is the catholyte, which it is the electrolyte that is placed near the cathode side. So the light yellow color is the catholyte. And fifth is the wireless tag. So this tag helps to transfer the information onto the mobile phones. So white color tag on the top is the tag. So in this thesis, the hypothesis was that if the soil is dry, there will be less bacteria that are present in the soil, and they will emit less electrons, and they will, because of the less electron, less electricity will be produced, and the less signal strength will be generated. When the soil is moist, there will be little bit more bacteria, more electrons produced, more electricity produced compared to the dry soil, and more signal strength will be generated. And when the soil is wet, there will be more bacteria, more electrons produced, more electricity produced, and the higher signal strength will be generated from the tags. So the first thing what we did is we designed the cathode. So why there is a need to design this cathode? Because the currently available electrolytes or the catholytes, they are toxic. They are toxic to the environment, like potassium ferricyanide. So we wanted to use the nitrate, which is beneficial to the agriculture, and they are environmentally safe. So for using the nitrates, we need the specialized cathodes. So we used copper and the nickel to design these specialized cathodes for nitrate reduction. We use physical vapor deposition technique to prepare these copper nickel electrodes of various thin films. So this is the machine. This is the device, physical vapor deposition, where the orange color copper is evaporated and get deposited onto the nickel plate. So in this experiment, we were able to reduce 10% of the whole nitrates. So these are some results, and we concluded from this is that the monometallic, like a copper nickel thin film, is not sufficient to reduce the nitrates from the water. So there is a need to use the bimetallic alloys, copper plus nickel, to design this cathode for effective nitrate removal. Then we designed the biodegradable anode cum membrane. So why there is a need to design these membranes? Because the currently available membranes are expensive, they are non-degradable, and they cannot be used along with the filter paper. So the polymers we used for designing these membranes are the chitosan and polyvinyl alcohol. So this is the cross-section of the whole paper batteries, like a membrane setup, where the top portion represents the anode, and below that you can see the sandwiched membrane, where filter paper and the polymers, they are absorbed. And in this we found out that the coated chitosan PVA membrane shows the lowest oxygen diffusion and the highest proton conductivity. So these are the results of these from this experiment. So the hash color shows the coated chitosan PVA membrane, whereas the blank graphs shows the membrane without coating. And all the membrane shows the slow degradation. That is, for 100 days they showed very less degradation. Then we decided to design the paper battery. So why there is a need to design this whole paper battery? So because we wanted to use the catholyte and in the liquid form. Then we wanted this whole assembly to be, it can be assembled, assembly should be very environmental, very easy for the common man to assemble. Then it should be compatible with the wireless tags. Next, it should have the increased surface area. So then we found out that the 3D cubical paper battery satisfy all these conditions, and we designed this 3D cubical battery by using the origami techniques. So this is the model that we worked, and this is the setup where we use this 3D model into the soil in the lab, where we use the soil bacteria that are present in the soil to perform these experiments. And we were able to produce, these are the graphs which shows the power density and the current density that we received, that we saw in these experiments. But there are some limitations in the future work and also the impact associated with this design. So what are the impacts? So the impact is that this paper battery will provide the off-grid low-cost sensing for the smallholder farmers in the developing countries. So it will help to reduce the e-waste, and it will improve the water use efficiency. But what are the limitations of this? The limitations are it cannot produce the high power, it cannot be used for the longer time, and it cannot be it cannot transmit the signal to the longer distances. And there are the scaling challenges because the soil types are different for the different regions. And the future work is that we need to optimize these electrodes for nitrate removal. We have to improve this design to increase the surface area, and we have to modify this whole setup to integrate the RFID tags. Till now we use NFC tag. We wanted to use the RFID tags to transmit the signal to the longer distance. So this is the future work. And after doing these future works, so we need to use the field trials, because without field trials, we cannot conclude that our project is successful or the prototype is successful.
Thank you very much for your presentation. So if you have the time, thank you so much.
Yes.
I would now like to start opposition, and I give the word to Professor Aravind. Please put on your cap.
Many director, many thanks. Vada Promovendus, congratulations with your thesis. It's a challenging topic and a serious effort. So, but let me just start to understand this work in the broader context. I have seen a couple of slides right now saying that, okay, well, there are challenges with water management and farmers should have devices to understand soil moisture content that might help them better. So there are many devices, right? There's a lot of data about soil moisture, how it varies, remote sensing opportunities coming up. So what are the other methods available to understand soil moisture, and why this work is super important?
Vada Promovendus, is this question clear to you?
Yes.
Then I give you the opportunity to defend yourself.
Yes. Who cleared the opponent? Thank you for the question. Why I will answer why this is important. I will talk about from the point of view of the India, like a developing countries. So as you know, in India we have smallholder farmers. They have like a very small farms on which they do the farming. So if we need to measure how much soil moisture is there in the soil, like you told me about from the satellite. So the resolution of the satellite is more. So they cannot give the exact information of that small farms. So it will be difficult for the farmers to use the satellite data. At the same time, the satellite, accessing the satellite data is not like economical to the smallholder farmers because they do not have the money to access this information. So that thing is not useful for the smallholder farmers. Okay, so why this is important? Because like the advantage of my device is that this will give the information about the soil moistures like at the near the near the crop. Like what our concept was that we will put this whole system when we will sow the seeds in the farm. So it will remain there for one crop season. So it will give the information about the particular crop, okay, and that too it is a cheap because the farmer just have to put this whole system into the soil and they can, and every farmer like nowadays every farmer they have the mobile phones like smartphones, so which are not 100% but 80% they are NFC enabled. So at least they can use these NFC enabled mobile phones to sense how much soil moisture is present into the soil. And as I told you, this will get a power from the soil bacteria. So there is no need for the external power sources like solar panels or batteries or electricity grid. So that problem is also solved with this device. And in addition to that, it is not making any it is not producing any electronic waste. So that is also the advantage. So yeah, so I answered the second one. Then the third is what are the other techniques through which we can sense the soil moisture? Yeah, there are some unscientific methods as per my knowledge. Like they use it's called touch and feel method. So they hold the soil into their hands and they sense the soil moisture. So and there are some radar techniques also. I think. So there is what they do is they roll there is a radar, they roll the radar onto the soil, onto the field, and then they can sense how much soil moisture is there. But these are the expensive methods for the smallholder farmers.
Okay. So thank you. So you see a role for this technology?
Sorry?
So you still see a role for this technology?
Yeah, yes.
Okay, good. Thank you. Well, can I continue with the questions?
You have plenty of time.
Thank you. Then a second question, when I listen to your presentation, but also I read your thesis, I read about or I hear about paper batteries, but also microbial fuel cells.
Yeah.
So are there things common in between? How much similarity you see? What are the differences? And also in the context of your work?
Yeah, thank you for the question. The similarity is, they have the similarity in the architecture. Like in the soil microbial fuel cell also, we are using anode, cathode, and the soil is it is not a membrane, but as an electrolyte. So in paper batteries also, they have this anode, cathode, and the membranes. So this is the similarity between the soil microbial fuel cell and the paper battery. So the only difference is that in soil microbial fuel cell, we are using the soil and carbon felt and other material as an anode and the cathode. But whereas in paper battery, we use the paper as a substrate on which they coat the various conductive inks, which are used as an anode and the cathode. So that is the only difference.
Could you please repeat and explain? Yeah.
So in soil microbial cell, we have anode, we have cathode.
Yeah.
In paper battery also, we have anode and the cathode.
Yeah.
So these anode and the cathode, they are printed onto the paper substrate in the paper batteries.
Mm-hmm.
Okay, so these are the similarities. Anode, cathode in the SMFMC, like a soil microbial fuel cell, and the anode and the cathode in the paper battery. So these are the similarities. So we wanted to use these similarities and combine these two technologies for designing our setup.
Okay, and what is your setup? It's a fuel cell or a battery?
Yeah, it's a combination of all these things. Like this is the model. So yeah, we can say it is made up of paper. So it is a paper battery. We are using in the soil to produce the current. So it is a part of the soil microbial fuel cell also. So it's, we can say it's there, it's a both.
Okay, okay. Well, let's continue. Maybe you'll have a. But of course, now one of the difficulties you have mentioned in your thesis is say calibration, right? If
Yeah.
you continue to use these devices, calibration is going to be challenging. Now, looking at your device and how it performs as an electrochemical device, so what are the challenges in calibration and why?
Thank you for the question. So, if we talk about the calibration, we are using the liquid electrolytes. So, using this liquid electrolyte and transporting this whole system to the field, it's a challenge. At the same time, these liquid electrolytes, in my setup, what I did, it was leaking from the paper battery. So that was the challenge. And another challenge is that we have the soils at we have different types of soil. So right now we tested only for the one soil that is from the Netherlands. But back in India, they have different types of soil and there are different types of bacteria present there. So that is a huge, as I said, it is the problem is with the scaling. So there will be various challenges in the calibration of the things.
But in the sense, you need to get a voltage and a current, right? How it's going to be affected?
Sorry?
You need to get voltage and current, and then that should actually tell you how much moisture is there, right?
Yeah, that is true. But the amount of bacteria that are present, they are also affecting the current production and the voltage production.
Okay.
So, like, if I am using black cotton soil, like in Maharashtra, we have the black cotton soil, and if we are doing calibration on that, okay. But if we use same setup in the soil, like in a red soil or in different soil, so the bacteria will be different. So the readings will not be same there.
Okay.
So we have to individually calibrate.
So what's the solution you foresee if you get into business?
So I didn't thought about what is the solution. Right now we were just focusing on to give a proof of concept towards the scientific community that, yeah, we can, because SMF is like a plant microbial fuel cell, soil microbial fuel, these are the emerging renewable energy technologies. So, we put a question in front of the researchers that there is a possibility to use these paper batteries and SMFC technology as a renewable energy source in the off-grid region. So we are just focused on the proof of concept.
Manager Director, can I ask one more question?
Professor Arvind, given the time, are you satisfied with the answers given to your questions?
Thank you. I'm happy.
Thank you so much for the opposition. Then I kindly invite Professor Strik to continue the opposition.
Thank you, meneer de rector. Waarde promovendus, congratulations with the completion of your PhD thesis. It was a pleasure reading it. And of course, I have some topics I would like to discuss with you.
Yeah.
But to start, I would like to ask meneer de rector, because I have a question about proposition number 5.
Waarde promovendus, could you please instruct your partner to read our proposition number 5?
Proposition number 5, yeah.
Like a kite in steady wind, innovators with all black or all white hair soar highest. Mixed hair makes the ride bumpy.
Yes. So, dear Sumit, can you explain me what you mean with this?
Yeah. Zeer clear the opponent. Thank you for the question. Yeah. So, why I wrote this proposition? Because I am 37 years of age. And the person who is at this middle age, so they cannot focus on one single topic because they have to take care of the families, they have to take care of other things also. So when you have all these responsibilities on you, you cannot focus more on your research. And why I'm talking about the all black hair on the all white hair, because when you have all black hairs, like you are very young, like you can say like 24 or 23. So you do not have these responsibilities. You do not have the family responsibilities. You do not have anything. So you can focus on such kind of a topics and you can dedicate your whole time onto that. And when you are too old, it's not offending for the old people. But when you are old, they have they cross all their limits. Like they do not have that much responsibilities on you because their children are young, like, and they can take the responsibility of their own. So they can also they can focus on the research more. So, but for the middle people, it's very difficult to manage all these things. So that's why.
Okay, that's clear. Then, like I'm getting some gray hairs, so I should watch out that that I'm not facing a bumpy road in the future as an innovator or scientist. Thank you for explaining that. Then I would like to start the first discussion topic, and this is the actual biodegradability of the sensor you developed. And in your thesis on page number 69, you introduce the biodegradation testing methodology, and here a kind of method was chosen. So can you explain me why this test method was selected?
Okay. Yes. Thank you for the question. Why I decided to do this degradation test in compost tea, because, as I told you, we are using these paper batteries in the agriculture field, and to grow the crops, the farmers, they use the compost. Okay, so the compost will affect the biodegradability. So in order to replicate the field conditions that the farmers are using in the, like, if I'm talking about from Indian perspective. So to replicate the same conditions in the field, we tested this degradation in the compost tea.
Yeah, so you want to get insights in the degradation, and I understand the role of compost because it's applied on the land.
Yeah, that's true.
But I was wondering whether, how comparable are the conditions in this test environment compared to an outdoor environment where such kind of sensor could be applied?
Yeah.
Do you see other potential differences there?
Yes, thank you for the question. Yes, there are more challenges if we test this setup in the field, because in the lab we have very less parameters that are changing, variable parameters. But in the soil we have different parameters, like the soil condition, the temperature, the pH we cannot control, then the temperature, and then if there is a sudden rainfall, there is a sudden moisture, increase in the moisture content. So there are differences. We cannot replicate totally, but at least we can replicate some parameters, like compost, percentage of compost, and temperature.
Yeah, and if you then looking at the results, then you also measured the degradation of the different membranes.
Yeah.
And let's say they're more or less degraded like 4 to 5% in about 100 days.
Yeah.
And it is discussed that a functional lifespan should be at least 100 days. So that could be suitable, I assume, because it's not fully degraded. But if you look now at your used method, and you would compare it to a real application, do you expect that under real application conditions the membranes would degrade faster or less fast, slower?
Yes, thank you for the question. So, like in a real condition, it will degrade faster. So, sorry, the next question.
So are you sure that it will be degrade faster under real conditions, and can you elaborate on potential mechanisms that are affecting the biodegradability?
Yeah, so as I told you, it will degrade faster because these membranes, they are the degradation happens because of the if there is a changes in the temperature, it will degrade because then if the bacteria they are changing, like other bacteria who are responsible for the degradation, they will also affect the degradation in the soil in the real field conditions. Then temperature I told, soil conditions. Yeah, so these are some of the parameters that will affect the degradations and in soil we will have more degradation.
Okay, thank you.
Yeah.
Yeah, then, but I was also thinking about this, the biodegradation of such kind of sensor, and I understand there's a kind of concern if you use sensors and they stay in the agricultural field that they can cause problems because the electronics could be still there.
Yeah.
Do you think it's possible to manufacture a sensor which is 100% biodegradable? Can you elaborate on this, whether that would be possible?
Yes, thank you for the question. So from my study, what I found out, and from the literature, the paper is 100% biodegradable, cellulose paper, because we are using the cellulose paper. But we are using these polymers. So chitosan is also fully biodegradable. But the polyvinyl alcohol is little bit not that much 100% degradable. So it is possible, like from the literature, what I found out that it is possible. I'm not sure it will be 100% but from what I used, they are 100% biodegradable. But the degradation time will be more. Because and our main focus was that it should remain intact for one crop season, like 4 to 5 months. Because in India we have the crop season of 4 to 5 months. So at least for 4 to 5 months.
And what do you think about the electronics? Because you have like the membrane, the electrodes, so they have a certain they can biodegrade. I understand this. This is also supported with your research.
Yeah.
But there's also certain electronics needed. In the lab, you probably use like a potentiostat, but probably something smaller would be needed if you would apply such sensor in a real situation. And you also propose to have a kind of device that the information can be transferred to a cell phone.
Yeah.
Is there potential to make these parts also biodegradable?
Yeah, thank you for the question. Yeah, so in our device, we used the electronic device. So we use NFC tags. So there are some companies like Identix, they are designing these fully biodegradable RFID tags. So that is degradable. Then we have the coating graphene also. So it can stay, but we can say it is not electronic part. We can use the cathode by printing these conductive rings or a copper nickel onto the filter paper. So that is also biodegradable. So the only thing is that that what I found out is the IC chip that we have in the NFC tag, but it is very small. So I don't think that it will affect the farm or the soil, because very thin, like we can say less than a 0.1 mm, so very thin NFC tags. So that is the only thing which is not degradable. So what? There are the wires.
Yeah, but it's small. But could it have an environmental impact in the end?
Yeah, but yeah, for short term, there won't be any problem. But if we are using lakhs of NFC tags, then there will be a problem, because we cannot replace the IC using any biodegradable materials. So, yeah.
Clear, thank you. Yeah. Then I would like to continue to another discussion point, and this is on page 102 of your thesis. There you show the current and power density curves of the experiment.
Yeah.
And let me see. And later on, you also show on page 104, actually, I want to first discuss this.
Yeah.
Figure 5.16. So there the chronoampermetry tests were done, and
Yeah.
The current is reported over time.
Yeah.
And different concentrations of acetate were applied.
Yeah.
So I was wondering, like at a certain moment, the current is somehow flattening off. It's somehow getting to kind of base current.
Yeah.
And in your discussion, you give several potential explanations why this is happening like this. My question is, did you also look into the coulombic efficiency of this system to better understand what could limiting the current?
Thank you for the question. So actually, I didn't do that in my experiments. So this was the last experiment. So like considering the time. So but we were much more focused on how much current we can produce. So we were not that much like Coulombic efficiency, how much electrons will be produced, because in our system we want only the electron flow that will interrupt with the NFC tag. So we were much focused on the electron flow, not onto the how much current how we can maximize the current.
Yeah.
So that was not our part. So
Yeah.
that's why we didn't do
Yeah,
that.
but maybe we can still look into the figure, because you supply different concentrations.
Yeah.
But I could not completely find out whether each, let's say, each spike had the same amount of total acetate, or whether different amounts of total acetate supplied.
Yeah.
There were different amounts in the
Yeah,
end.
different amounts. Yeah.
Okay. So then, do you see there a correlation between if you supply more, there's more current, or not?
Yeah, so like when we see from the graphs, like after 0.5, there is like going a little bit increase, but there is no certain trend we found out from this. Like as we increase the amount of the acetates, there is a linear relationship between the acetate. So at least in our experiment, we were unable to find out, but I do not know about the others.
Yeah.
Professor Streeck, given the time, are you satisfied with the answers given to your questions?
Certainly satisfied with the question. So
Then thank you so much for the opposition. Then I kindly invite Professor van Lier to continue the opposition.
Dank u wel, meneer de rector. Waarde promovendus, also from my side, congrats with the accomplishment of your thesis that I read with great pleasure. Well, in your thesis, you are going for a remote moisture sensor fueled by microbial oxidation, either from root exudates or other kind of soil organics. And while finishing your thesis, I think, well, it's still far from realizing the sensor, I realized. And also in your research, you pointed at different limitations. And I would like to discuss some of your limitations. You just showed an example of your, maybe you can show it once more. So this is a very small device.
Yeah.
And for your microbial fuel cell, or your battery as you call it, you need an oxidative cathode, right? So you need oxygen as well to make for your reaction, or at least an oxidized environment. Not everything can be reduced. So, and that means, but this is at the top layer of the soil. Now I learned when I was working at Wageningen a long time ago that if you have a water stress agriculture, that plants are rooting deeper. At least a farmer would also like to root deeper. So do you feel that if you sense then the first centimeters of your top layer with this device, that you can predict also your moisture content of the deeper layers? Particularly if you have a high temperature, your transpiration is, of course, higher at the top layer.
Who cleared the opponent? Thank you for the question. So thank you for the question. So as I told you, like in the future work, so what we are thinking is to embed the RFID tags. Right now we are using the NFC tags. So the range of these NFC tags is like 10 centimeter. So we can sense only the top 10%. But if you want to go deeper, we have to use these RFID tags, because these RFID tags then can emit the signals to longer distances, like 100 meters. So with those RFID tags, we can go deeper into the soil, but not with what I did, like with the NFC tags. So it is not possible.
Okay. Okay, fine. That's fine. Then the other limitation is the moisture content itself. So moisture is triggering your electric exchange of electrons, microbial exchange of electrons. What is the minimum moisture content that you need for microbial activity?
Yes, thank you for the questions. So like what I found out is that these bacteria from the literature, 30% to 70% is the optimum soil moisture where they can grow. Below 30%, they will die. And if the moisture content is more, so for some bacteria, it's not good. So the optimum range is like a 30% to 60, 70%. I do not know the exact, but 30 to around 70. This is the range with which we can sense the, like how much soil moisture is there.
And
30%.
does that also trigger the need for irrigation? Well, I'm not an irrigation specialist, but is that the same range that you need irrigation for your plants?
Actually, I do not know the exact thing, but at least what information is we can get that if initial soil moisture is 60%, if the initial soil moisture is 60%, and if the soil moisture is decreasing to 50% or the 40%, then the farmers can get the alarm that yeah, the soil moisture is decreasing. Then from his intellect, he can decide that yeah, we have to irrigate the farms or not.
But it depends also on your soil types and so on. You also mentioned in your layman's talk, okay, soil type difference when you have more organics in the soil or no organics in the soil. That will also how severe is a certain moisture content of a soil is, I expect, right? So this direct translation has also, yeah, it will be impacted by the type of soil that will be irrigated and the heterogeneity or homogeneity of that soil.
Yeah, that's true. Like, but I will talk about the other part of these soil moisture sense. Like, when I told you that these farmers are using traditional irrigation practices. Like, if they know, like, right now, what is the soil moisture that is present in the soil, that is 60%. So at least they can decide that now we have to use only like a 20% of the water. So they will not overload or over irrigate the farms, which will result in the water loss. At least they can decide on this. So yeah, so in this way they can get the information.
So that means they will have a kind of a personal adaptation period, calibration period of some seasons in order how to interpret the data of your device.
Yeah, thank you for the question. Yeah, that is true. But yeah, like this, as I told you, this is like just like a initial stage of developing this kind of a sensor. So in future, if there are the like exact relationship between the soil moisture and the current, then they do not have to use their intellect that how much soil moisture, how much water they have to put to the farm. So right now we do not have. So for the initial, they have to depend on their intellect. But in the future, as the science progresses, they can directly decide by looking at the graph or by looking at the results what they are seeing on the mobile phones.
And what do you expect between the soil moisture content and the signal that will be produced?
Yeah, so there is a direct relationship between the soil moisture and the signal strength.
How is that relation?
Yeah, this soil moisture, bacteria in the soil, there is a direct relationship onto the soil moisture. If the soil moisture is more, there will be more bacteria.
Wait a minute. Yeah, you have soil moisture, and that then you have bacterial activity.
Yeah, that's
and then in the bacterial activity, oxidation of root exudates.
Yeah.
You generate electrons, right?
Yeah.
But do you expect, if the soil moisture is increasing, like a linear relationship with the signal that you're going to produce?
Yeah, so like...
or what type of relation you expect?
Yeah, as I told you, there will be for the 30% to 70%, there will be linear relationship. But if...
Linear? Why linear?
As I seen in the literature, like they found out the relationship between the soil moisture and the bacterial activity. So I do not know exact whether it is a linear, but there is a relationship between the soil, soil moisture and the bacterial activity.
Bacteria has an exponential growth rate. So if a bacteria is in a dormant stage, and suddenly there is more moisture coming up, so there's more exudates are present, then you have a wild growth of bacteria which follow Arrhenius, exponential curve, I mean. So why this linear relationship?
Yeah, so you are saying that the bacterial, so when these bacteria will grow in larger amount, when there is a more soil moisture. So there is a direct relationship.
Yeah, but not linear.
Sorry, mistake. There is a relationship, direct relationship between the soil moisture and the bacterial activity. Not the linear, But
Okay.
there is a relationship.
Okay, fine. So the higher the strength, the more moisture. So
Yeah.
the lower the strength, there will be a trigger for
Yeah,
irrigation. That's the idea. Okay, and imagine so when there is more moist in there, it's not only the electrogenic bacteria that are there, but there are also other type of bacteria. Rhizobium, Azospirillum, Bacillus, capturing the nitrogen from the air. If these bacteria are around your device, what will happen?
Yeah, that's a good question actually. For this, there is a one technique called the quorum sensing. So using quorum sensing, we can attract only those bacteria that we want to use. So there are volatile organic compounds. So we can use those volatile organic compounds to attract the bacteria that we need and who are exoelectrogenic. And then we can keep 100%. We cannot keep the other bacteria away from our device, but we can invite the more bacteria to come towards our battery. So that is the future work. So we put these volatile organic compounds into the ink to attract those bacteria. So that is the future work. So we didn't thought about that.
That sounds a little bit quite far away. Still, quorum sense to use quorum sensing for this type of appliances, but I guess okay. But there will be a direct competition with your electrogenic bacteria, right? So.
Yeah.
So your signal will be less, despite the fact that you have a higher moisture, which leads to misinterpretation.
Yeah, that's true. So, yeah, right now we are not focusing on that. So we are focusing on the current production. But yeah, there is a still study need to be done to how we can keep those other bacteria away so that they do not interfere with our exoelectrogenic bacteria. So there is a need of, there is a scope for doing this research.
Dear Director, there's a room for one more question.
Professor Van Lieren, given the time, are you satisfied with the answers given to your questions? Then I would like to thank you for the opposition. Then I kindly invite Professor Uilenhoed to continue the opposition.
Dank u wel, meneer de rector. Waarde promovendus, I also read your thesis with a lot of interest. It deals with a societally very relevant subject, scientifically and technologically also quite challenging, as we have already heard. I have a few questions, of course, which I would like to discuss with you. And my first question pertains to, let's say, the placement of your work in the field of, let's say, science and engineering. Looking at the papers that you published and the journals where you publish your papers, they are not the typical journals, let's say, where I publish my papers. That is all fine, no problem with that. But they are not, let's say, typical journals in the field of, let's say, agricultural water management, right? They're focused on, let's say, the sensing technique, actually, that you have developed along the course of your PhD thesis work. So I was just wondering, where do we place your work in the field of science and technology?
Thank you for the questions. So yeah, you are true. This is a multidisciplinary project. So we cannot target any specific. Right now, we do not have any specific journals or a specific science, because being a multidisciplinary, we can publish papers in any journals. So right now, what I think is that from the work which I did, it's basically a part of the material science. Because we are developing cathodes, we are developing membranes, we are using the paper batteries. So we can say like not 100%, but at least I will give advantage to the material science, like 60%, that it will belong to the material science department.
Okay, thank you very much, because that gives me an idea of where to put your work in, let's say, the entire range of disciplines that we cover here at TU Delft. Now, suppose you would get your sensor that you show here to work. What would be, let's say, the representative soil volume that it would cover, ideally?
Yeah, that is a good question. The range of this our prototype will be depend on the dependent on the bacterial activity. Like if they can sense and get settled down onto this electrode, like from 5 centimeter or 10 centimeter, then this will be the range for that. If beyond that, because there is no relationship between these, like those are not affecting. But if the bacteria are coming from 10 centimeter, like from future experiments, then we can say that the range of this is 10 centimeter. But if we use chemical like volatile organic compound and we increase, we are able to attract these bacteria from 15 centimeter or 20 centimeter, then the range of this battery will be 20 centimeter. So it depends on that thing.
And what are the factors, let's say, that affect that distance?
Like the water content is the one factor. Then we talked about the root exudates that are present, because if the root exudates are in the vicinity of this paper battery, so we cannot tell any exact figure that what will be the range. But we can guess that if the root are exceeding to 10 centimeters, then at least there is a possibility that we can sense till that. But right now we do not have the exact number that this is the range that we can sense the soil moisture.
And that is also affected, if I understand correctly, by, let's say, the soil moisture content itself.
Yeah.
So there is some non-linear effect there, in the sense that, you know, the variable that you try to measure has a certain variability in space and in time. But let's say the sampling volume that your device would actually capture, so to say, is dependent on the variable you're trying to measure in the first place.
Yeah, that's true.
That seems to be still then sort of an open question that would be interesting to study, let's say, in upcoming work. Yeah, thanks. So, and you know, why soil moisture in the first place, right? You also in your thesis, you start with the problem of agricultural water management in the global south. You give the example of India, obviously, and then, you know, quickly you, let's say, bridge, so to speak, or you make this leap towards soil moisture, right? There would be other, you know, quantities or variables of interest a farmer, you know, could focus on. Why particularly soil moisture, and which other variables, you know, are at play, so to say?
Yeah, thank you for the question. So as we know that the primary thing that is required for the crop growth is the water. Like without water, plants cannot grow. So that should be the main. Like then we can add up on to the nutrient content or like other things. But the primary thing is that we can neglect the soil also, because like in hydroponics we do not use the soil. We do not use the soil. So but the water is the important. So whether it is like in dry soil, there will be less water, but there is a water. So that should be that is the primary thing that is required for the agriculture sector.
So it's about water availability.
Yeah.
Clear.
Yeah.
But let's say the plant itself, or the agricultural crop, the status of the crop, to a certain extent, is also reflecting whether or not there is, you know, enough water available or not, right? So would you think that, you know, focusing on, let's say, the status of the vegetation itself, the crop, you know, for instance, the way in which it's able to evaporate, could be another, you know, perspective to, let's say, the same general problem, or could provide complementary information?
Yeah, so what I think is that it will be a complementary information, because the farmers right now, like some of the farmers, they see like if the leaves are drying, they think that there is a less soil moisture. So based on that, what I see is that it can be a complement to the farmers, because then they can exactly say that yeah, they are drying. They are dying because of the soil moisture, because like these crops, the leaves of these crops, they can get damaged because of the diseases also sometimes. So that is not the problem of the soil moisture. So then the farmers might get misunderstood that the leaves are getting damaged or getting dried because of the soil moisture. So if the farmer get the same information from from our device and they see that something is happening to the crop, then he can take the exact decision about the soil moisture.
Clear. Thank you very much. And so I like that idea of complementarity.
Yes.
And in a sense, that would also hold, I think, for the other devices, so to speak, that are available, that are around to detect soil moisture. One of the other opponents mentioned remote sensing. And your response there was, well, but remote sensing provides soil moisture information at best at a relatively coarse scale, which is not well adapted to, let's say, the individual field of a farmer. My device, is going to provide, local information. Do you see a potential there for those two, let's say, sources of information to be combined to provide better estimates of soil moisture, or let's say water availability?
Yeah, so thank you for the question. So I will again go with the complementary answer because if right now the satellite, I do not know the exact resolution of the satellites. If the satellites are more advanced and they can give the information about like at a very low resolution, then the information from the satellite and the information, then it can complement. But right now, the satellites, they give the information on a larger scale, not for the at a very small scale. So right now, my device is perfect. So in future, if the satellites are more coarse, they can sense the resolution is higher, or we can say that they can sense more, then it can be complementary. But right now, this device can be the perfect for measuring the in situ moisture.
Thank you. And so how do you see this in practice? So suppose, I don't know, a year, 2 years from now, 3 years perhaps, you have a working sensor that does what you want it to do. And are you going to, let's say, distribute these sensors to farmers, like one sensor per farmer or one sensor per field? Or how do you see this in terms of, sampling, the variations that you have in practice in a field or in a river catchment or in a province?
So as I told you that right now we haven't decided the range, that till what range it can sense the soil moisture. If we know the exact range at like 10 centimeter they can sense, then we can decide that how much soil moisture, how much these devices are required for each field. So right now we do not have the exact range. So when we find out that this range is perfect for this, then according to the area of the field, we can distribute these sensors to the farmers. So that is the question, the open question for the scientific community, that to find out the exact range that it can sense.
And in terms of investment, I mean, what are we talking about? Because you're saying, let's say, a sort of a sphere of influence of tens of centimeters. I don't know, maybe a meter. If you would, want to cover all of that variability, you need, hundreds, thousands of sensors per field. Is that realistic, given, the biodegradation on the one hand? Is that realistic from the, let's say, the cost perspective on the other hand?
Yeah. So right now in India, so the government is encouraging.
Thank you, Madam Biedel. I request the promovendus and Pagenim to take their seat again. The committee will withdraw for further deliberation in the committee room. The session is adjourned.
Okay, this. Ah, okay.
You will come to the office after?
Yeah,
You will see around there.
but thank you for you guys. Really helpful, yeah.
What your supervisor and other guys said today? Today what he said?
And I see the sense.
Huh? No, it's not like it's a prototype. It's not working.
But I would love to see.
He knows that what are the limitations. He was stating from the start. But without these guys, we will not be able to finish. Many thanks. No, no. We have two sections. One long student and one is second. We have two.
It's like you spend like 5 minutes and then 10 minutes to present.
Yeah.
That's like,
I think I was over.
not,
Not that what we did
not
wrong.
2 minutes. Like.
Okay. Explaining all these things. Without that, the common people will not really understand. But what I'm doing, if I talk directly about paper like
If your supervisor discuss what he's going to speak now, did he discuss anything?
right now,
Yeah, after the ceremony, like when they award your degree, he has to talk 15 minutes about
I do not know what he's going to do.
That's really interesting.
Yeah, I trust him.
Second person is a co-promoter, right? The one
Yeah.
who is the last one. He's the promoter. That's the one next to him.
He's promoter, that is supervisor.
I think the promoter will read it.
The one who was actually doing that.
Promoter will sign, but he will be signed.
So sure. outskirts or like within the city?
No, earlier my area was outskirts.
Okay.
Now it comes to the city.
So you now really crowded.
Yeah, no, it's very crowded. In 2000, the government came to draw because of the political pressure. It's still the land is not in our name.
It's quite common.
When they came, all the people they shifted their all the things to like new places. It was a big problem. Because right now it's not in our name. So he used to stay near my house during his master bachelor studies. Yeah. And little bit hesitating like whether to invest such much money, lot of money. Unless I think we have to start by beyond.
I reopen the session and request the promovendus and Pagenhif to stand in front of the committee. Het College voor Promoties van de Technische Universiteit Delft, vertegenwoordigd door de hier aanwezige commissie, heeft, na kennis te hebben genomen van uw proefschrift met stellingen en naar uw verdediging daarvan te hebben gehoord, met inachtneming van de bepaalde in de wet op het hoger onderwijs en wetenschappelijk onderzoek, besloten u de graad te verlenen van dokter. Ik verzoek de promotor, door het College van Promoties als zodanig aangewezen, zich van de hem opgedragen taak te kwijten.
Uit kracht van de bevoegdheid bij wet toegekend aan het College van Promoties, verklaar ik als promotor namens dat college, hier vertegenwoordigd door directeur Magnificus en de overige leden van de commissie, bij deze u, Sumit Maya Moreshwar, te bevorderen tot dokter en u alle rechten te verlenen welke aan de dokterstitel zijn verbonden. Ten bewijze hiervan overhandig ik u het diploma dat u het recht geeft de titel dokter te voeren, ondertekend door directeur Magnificus en de promotoren en co-promotoren en voorzien van de zegel van de Technische Universiteit Delft.
It is the co-promoter's privilege to be the first to congratulate the young doctor. Professor Pande, the floor is yours.
Thank you, Meneer Director. Thank you, Meneer Director. Dear Dr. Meshram, dear Sumit, congratulations. Not only from me, but also from Ludovic and Nick. We are delighted to have been part of a journey that culminated in this nice defense today. Your dissertation investigated whether a soil moisture sensing solution exists that not only performs effectively, but is also cost efficient, operates independently of conventional power sources, and minimizes environmental impact by naturally degrading over time. In this journey, you fabricated copper-nickel bimetallic electrodes using physical vapor deposition. You synthesized a cost-effective biodegradable membrane separator utilizing chitosan that then you used to design a 3D and 2D paper battery. Your dissertation finally provided a proof of concept of working paper fuel cell batteries with novel foldable origami way anode and cathode materials. The novelty of your approach is evident. No one has thought of using microbial fuel cell inspired paper batteries, not to power off-the-shelf sensors, but to sense soil moisture directly. Only if you had more time, you would have provided a proof of concept of how such a nature-powered soil moisture sensor can detect soil moisture conditions during a crop growing season. This takes me to the many challenges that you have faced. Before you started your PhD journey, you were a water engineer who was very motivated to solve water challenges of farmers in Maharashtra, India, being yourself from such a community. This was evident from the very first time you contacted us with your interest in pursuing the journey. You started with no training in biogeochemistry, electrochemistry, or material sciences, not to mention any knowledge of alien-sounding words such as potentiostats, metallic vapor deposition, or other lab materials that you've had to procure. You persevered in the face of these challenges, often surprising your supervisors by setting up successful experiments that proved that the new electrode materials and battery concepts actually work. You showed your bravery and success diving into and sorting out bloody experiments and its data, and working with challenging biotechnologists, material scientists, and water engineers. You have now matured as a serious researcher who now knows how to ask the right questions and how to learn new skills needed for the task at hand on the job. What I admire the most is the deep calm with which you have tackled all the challenges and uncertainties. I'm sure with this nature of yours and skills that you have developed during your PhD journey, that you're well set to make significant contributions in research or to start a world towards sustainable, nature-powered, environmental monitoring solutions.
Learned Dr. Moreshwar Meshram. You now have the right to use the title of doctor. Your doctorate means that society can rely on your judgment, that you will act transparently and communicate independently about your results and the societal relevance of your work. In other words, your doctorate implies that you will uphold scientific integrity. I wish you a great deal of wisdom and prosperity with your new status. On behalf of the Board for Doctors of Delft University of Technology, I congratulate you on earning your doctoral degree. Dr. Moreshwar Meshram, you may return to your seat. Ladies and gentlemen, I would like to thank you for your presence here today. I hereby close this academic session.